Hi everyone, this is going to be the second part of Progress Assessment 2. And I am going to specifically be talking about the process of plant evolution and conserved biochemical defenses between the Marchantia liverwort and the oomocyte pathogen listed here. So within the article that we were given, this plant evolution article, I was specifically interested in this section here that talks about the diversity of protection mechanisms in plants. And it says the dynamics of plants defending against microbes and pathogens impose strong selective pressures that result in rapid adaptive evolutionary changes. And in the bottom of this section, it says it's supplemented with plant lineage specific, but often conceptually convergent strategies. So I wanted to look at that a little bit more in this presentation. And this specific oomocyte pathogen I'm looking at, it's basically known as a water mold. And this group includes several hundred organisms that have some of the most devastating pathogens that we see. They are filamentous and heterotrophic, and they can reproduce both sexually and asexually. So for this, I looked at this article here, and they wanted to see conserved biochemical defenses between this oomocyte infection and then early divergent plant lineages. So specifically, I looked at phenylpropanoid-mediated biochemical defenses, and it found that infection of the species listed here results in the upregulation of these metabolism enzymes. So upregulation is an increase in the number of receptors on the surface of target cells, making the cells more sensitive basically to like hormones or other agents. And it includes those involved with flavonoid biosynthesis. So flavonoids protect plants from different biotic and abiotic stressors, and they also act as a UV filter. They function as signal molecules, allopathic compounds, detoxifying agents, and antimicrobial defensive compounds as well. And this particular kind of biochemical defense was found to be macroevolutionarily conserved between plant lineages, which I thought was super interesting. And the article also said that genes here show similar induction patterns in the liverwort itself and the angiosperm. So we haven't talked about angiosperms in class a lot yet, but I think it's really interesting to see how different plant lineages share similar traits and, kind of shared some of the same mechanisms to protect against these pathogen infections. So the article found that pigmented flavonoids, such as anthocyanins, are widely distributed in angiosperms. And that basically contributes to the flower colors that we see. That's why they're so diverse. And they usually protect against outside stressors by acting as antioxidants. And so when they're stressed, liverworts form this red-colored pigment known as richidin from this Marchantia genus. And it basically turns them red color. Color changes can occur from an accumulation or loss of these anthocyanins or an accumulation or loss of carotenoids in flowers. So anytime you see a change in color or anything like that, it's probably from either an accumulation or a loss of one of these two things, which is super interesting. And flower color changes can also result from pH differences. If you change the pH of the soil, that'll sometimes change the blooms that occur in a plant. And it's really interesting to see how these metabolic processes can change something like the color of a plant when it's stressed or being affected by something else. So also in the article, it says that these aronidins replace anthocyanins in bryophytes, and that anthocyanins were not present in the last common ancestor of bryophytes and tracheophytes. So it's really interesting to notice how this convergent evolution seems to be happening between these lineages. And even though these aronidins are not present in their last common ancestor, they replace these anthocyanins in bryophytes. So I think that's really cool. So for the figure that I want to interpret, it came from this article, and there's definitely a lot of information here, but I think it's really good to look at. So there's a couple different sections. I specifically want to look at this A panel right here, and it basically reflects hierarchical clustering of differentially expressed genes during infection. So you can see the infected ones on this side and the mock ones on this side, and then up here, these numbers represent the time points post-inoculation. And then on B, you also have some volcano plots that shows differential analysis per point in time. And then any of the genes that are significantly expressed are shown in the color red. So anything on the outside is significantly expressed. And then C takes upregulated and downregulated genes and compares the two of those. And then, you have D and E, which are a couple different protein groups where they're enriched, where they're depleted. And overall, this is just showing how this specific genus of liverwort is being affected when it's infected with this water mold pathogen. And I really like this figure. I think there's a lot of information here, and it kind of is just interesting to see how it's affected by it and how much control a pathogen really does have over gene expression. Overall, this article found that these pathogen-responsive orthologous genes with roles in trafficking, antimicrobial defenses, transcription control, cell signaling, and stress-associated metabolism. Basically, all of these genes have all of these effects here, it's a lot. Cell signaling, stress-associated metabolism. And then they also found that there is a conserved role for these phenylpropanoid defenses in mitigating pathogen infection in liverworts, which presents as an ancestral layer of the plant basal defense response. And that was likely critical for the expansion of plants onto land. So so essentially the expansion of plants onto land basically began the evolution of defense strategies to protect against these microbial invaders. And there's both a transcriptional response and a proteomic response of the early divergent liverwort marchantia with the pathogen that we discussed here. And basically, the results from this article found that the responseless infection is evolutionarily conserved, and there's an ancestral pathogen deterrence strategy that's centered on this phenylpropanoid mediated biochemical defenses. And I just think it's really interesting that things like this remain consistent across plant lineages, and it's definitely something interesting to consider as we look back on the evolution of land plants and how different mechanisms are all interconnected between land plants.